we've got a question. If we can't isolate the contribution of offshore wind farm to chemical release due to the other major source, how will it be possible to investigate chemical accumulation around offshore wind farm in sediment and biota and attribute it to offshore wind farm? I guess, Christelle, you can talk about the chemical tracer, perhaps?
Yeah, this is a big challenge in the future, is to distinguish the inputs from the natural environment and for the natural sources from the offshore wind farm. So it will be very important to develop some process and tools to trace these elements, and we propose to monitor the elements such as indium or gallium that are considered to be principally come from the anodes. So we have to see if it could be pertinent for the future tools to develop.
Yeah, just to complete the response, indium and gallium as targeted during the not Anemoy project, but Off-Chem project and the publication of Riz et al. And they try to follow for offshore wind farm in Germany, in the North Sea, the activity of the offshore wind farm with these elements. And as we mentioned, ECCP is increasingly growing for cathodic protection. So perhaps Jean-Luc, you have some target element to trace the activity of offshore wind farm focused on the cathodic protection?
Yes, what I said, it's about bromoform. Bromoform is known to be produced not only by wind turbine, but also by algae. So it's important to discriminate also bromoform from natural source to anthropogenic source. It is possible to discriminate it by isotopic analysis, by analysis the ratio carbon-13, carbon-12. It's possible to discriminate the source of bromoform, for example, but the other chlorination byproduct are able to be discriminated also by isotopic analysis. Mm-hmm.
And isotopic analysis was also proposed for trace the activity of offshore wind farm, considering the trace element of some anode. I don't remember the name of the element in English, sorry. Le plomb.
Lead.
Ouais. Sorry. Okay, another question. Are you able to put the concentration of chlorination byproduct compound from ECCP system into context for us? How high are the levels since here compared to what would occur naturally? And do you have any first thought on their toxicity for marine life? I guess, Jean-Luc, you almost respond to this question. And Christelle, you mentioned during your conclusion some ecotoxicological assessment for the cocktail of ECCP. Perhaps you can detail this question.
What readings? Jean-Luc?
Maybe I can answer better to the first question from Hélène. What I see in literature, it's at the level of bromoform, for example, generated by algae is very low as compared to what we found during our experiment. The level of bromoform generated by algae is around nanogram per liter, and the level of bromoform generated during our lab experiments is around microgram per liter. So the level is higher when it is anthropogenic. As regards the toxicity of DBP, CBP, yes, there are some literature on this compounds. Yes, I have not presented it today, but there is some data. There are some data on the toxicity of chlorination byproduct.
And just to complete this response, during the ECOCAP project, we also perform a chemical risk assessment for the main chemical from ECCP. And for some of the chemicals of the chlorination byproduct, some PNEC exists, but the assessment factor applied to calculate, to estimate this PNEC for this chlorination byproduct are very, very high. It's almost only translation from, but not translation, but use of ecotoxicological data from freshwater bioassay. So I think it's a big thematic for the future to enhance, improve our database on the chlorination byproduct in marine environment. And there is another. Sorry, Christelle.
Maybe
yes.
to complete your answer, your call, if we have to consider all the molecules released from the both cathodic protection, ICCP or GECP, the challenge is to perform is to know all the molecules released. So it's not a real problem for GECP because we know the metals, the elements, the different elements in the composition of the initial node. But for the ICCP, we, as we said, you said it's we did not have all the molecules released from the ICCP, and we did not identify all these molecules. And if you want to have an answer for the cocktail of these elements, you need to perform experimental, ecotoxicological experimental, also exposure of living organisms on the different molecules separately. And after we need to have the answer for the cocktail of these molecules. So it's difficult if you don't know all the cocktail, all the molecules constituted this cocktail. So I think it will be a challenge for the ICCP experiments that we can perform for the future ecotoxicological assays, bioassays. I think I don't know if I was clear. I hope.
Thanks, Christelle. Just to conclude our webinar, we will respond to the last question from Quentin Vertrez. For the effect on the biota, will your future project include bioaccumulation experiment along the food chain with local species, especially with cocktails? Actually, during the ECOCAP project, we develop this question. Christelle, you can perhaps talk about all your analysis on bioconcentration. So here, just bioconcentration.
Yeah, we try to choose this local species as we can work with these local species in controlled conditions. So it will be the first step. It will be the first choice. Sorry, it will be the first choice to select local species. And as I said before, the challenge will be to expose these species to all the molecules included in the cocktail, and separately to the individual molecules. So we need lots of experiments to have a complete answer of and the results for these experiments.
And to complete also during the ECOCAP project, we develop a tool to process some modeling approach in trophic contamination of GACP element. We focus our protocol on aluminium in the Gulf of Lion area, and the idea is to develop this tool for other area, other chemicals, organic or inorganic. And to respond to the question of the bioaccumulation experiment for future project, actually, yes. The ECOCAP project, we will try to continue this thematic with new project. We are in the research of founding now, and the question of bioaccumulation. So the trophic way, considering the trophic way, is also considered in our main future topic. For the speaker, is it okay for you, or you want to?
Okay.
Okay, so we will close the webinar on the ecotoxicology of cathodic protection. Thanks a lot for your participation. Very, very great thanks for all the speakers. Thanks Emma, thanks Christelle, thanks Jean-Luc, and thanks Nicolas. Thanks also Ronan to help us to perform a good webinar, to have a good condition to perform our webinar. This webinar will be available on the YouTube channel of France Énergie Marine in the very next day or the very next weeks, so soon. If some question remain you after the webinar, you can ask France Énergie Marine, and we will try to respond with the help of the speaker of the webinar or the consortium, help of researcher of the consortium. So thanks a lot. Christelle, perhaps you want to say a word as a principal investigator of the ECOCAP project?
It was a big adventure. It was very interesting, and I am so I'm happy of the results, the first results that we get from ECOCAP, and to share this adventure with all the partners, especially them. Thank you.
Thanks. Bye.
Bye. See you.
Thank you. Bye.
Thank you. Bye.